Hi guys, I'm so sorry I'm not there today, but I've already heard from Mr. Metz, and he said you guys were doing great, and you guys were great classes, so I really appreciate that. It's kind of stressful as a teacher being out, especially unexpectedly, so it's really great to hear that you guys were doing what you needed to do. So again, thank you so much. So we have to dive into cell parts. So there are specific parts or organelles inside of cells that help the cell to function. Not every cell is going to have every single one of these parts. We have bacteria cells that don't really have a lot going on. They're very simple. And then we have our more complex eukaryotic cells. So pause at any point if you need to. We're not going to go through all of them. I will save some for tomorrow just because it could be a lot. But pause, take a break if you need to, and we'll kind of keep pushing our way through so that we can be at a good place for tomorrow. All right, so let's get started. We're going to start with the four, and hopefully you have your chart, so the four structures that every single cell ever, no matter how simple or complicated, is going to contain. So it should make sense that every cell is going to have a membrane around it. It's the first thing that someone draws when you ask them to draw a cell. They kind of draw a circle. That represents the membrane or the boundary between the cell and its environment. And it's really important because not only does it surround the entire cell, so picture like a bubble in the air, or maybe a tennis ball, right? So you know how that green fuzzy stuff just covers the entire thing. Imagine that is the membrane. And inside is all the stuff of the cell, the really important stuff that we need to keep separate from the environment. So it's surrounding the entire cell. And it's what we call selectively permeable. So it's really important because the cell is constantly exposed to its environment. So even your cells in your body, they're exposed to their outside environment. And certain materials are going to need to enter the cell or exit the cell. So all cells really need some of the same things. We need water, we need oxygen, and we need nutrients or food. So that's going to need to come into the cell. And then waste and CO2 are going to need to leave the cell because we wouldn't want those waste products building up inside of it. It's toxic. So they need a way to exit. And the cell membrane, or the plasma membrane, is going to be the way that this stuff either enters or exits. As you can see in our little diagram down here, it's made of phospholipids, our super special lipid. And they're special because, unlike most lipids, they have, in addition to carbon, hydrogen, and oxygen, they have a phosphate or phosphorus added to them. And it gives them the special property of this little yellow part, the head, loving water because it's polar, so we call it hydrophilic, water loving. And it has these two fatty acid tails attached to each one, and those are nonpolar, so they are hydrophobic, which means water fearing. And guys, they are not just going to fear water. They do not like anything that is charged. So not just water, but we just like to call it water fearing or hydrophobic because it's easy to remember. And this is going to be found again in all cells. So whether it's a teeny tiny little bacteria or it's a much more complex plant or animal cell, we need to have a cell membrane to control what enters and exits. So that is the first thing. The second of our very basic structures is the cytoplasm. So if you look in this diagram up near the top, it's depicted as blue, and it kind of looks like empty space, but it's not empty space. So I want you to picture in your head jelly or jello. It kind of has that consistency. And what it's going to do is it's going to help hold those organelles up. So they're not just floating in air. They don't float in air. They kind of float in this jelly-like cytoplasm. So that's really important. And it's also the site of the cell's metabolism. So we need to remember that metabolism is all of the chemical reactions that are happening inside of the cell. So most of those chemical reactions are going to be happening right here in the cytoplasm. And then that must mean, then hopefully we're thinking about this, that the cytoplasm contains lots and lots of enzymes, which, remember, are proteins that speed up or catalyze chemical reactions. So if all the chemical reactions are happening here, it must make sense that we have lots of these enzymes that work with those chemical reactions here as well. So here's your fact you can write down. Cytoplasm contains a gel-like fluid called cytosol. You are totally allowed to use cytoplasm and cytosol interchangeably. You'll hear me do that all the time, just depending on which word comes to mind at the moment. But it's kind of 6 in one, half dozen in the other. It's the same. And this, again, found in every single cell. So no matter what type of cell you're looking at, everything has to have cytoplasm. The third of our 4 basic structures is DNA. And you see this fancy word up here, chromatin. It's, again, just a fancy word for DNA. And we've talked about DNA briefly when we talked about our 4 biomolecules. Remember, we have carbs, lipids, proteins, and nucleic acids, and DNA is one of our nucleic acids. DNA is found in all cells. And in your body, this is a common misconception, every single one of the cells in your body contains the exact same DNA as every other cell in your body. So I just want to make sure that I say that because some people believe that different cells contain different DNA of the same organism, and that's not true. So your DNA contains all the instructions to make you who you are. And how it does that, because remember, this is happening in every single one of your individual trillion cells, is that the DNA is telling the cell what proteins it needs to make. And this can translate then into physical manifestations. For example, eye color, hair color, and skin color all have to do with your pigments that your body, that your cells produce, which are proteins. So someone who's very fair-skinned, like me, I don't have the instructions to make a lot of the protein melanin. I just don't have it. My DNA doesn't tell my body to do that. Someone who has much darker skin, they make much more melatonin. It's the same protein. It's just a difference in the instruction. So when we talk about proteins, it actually does a lot of times manifest as a physical characteristic. And we'll talk about that when we get to genetics. All right, so our fact is they're made of the monomer nucleotides. Remember, monomers are building blocks. And this should say all living things build their DNA with these same 4 nucleotides. So we only have 4 options for those building blocks. And it should kind of blow your mind that we can then build all the living things on Earth just by changing those building blocks, like the order of the building blocks and the order of the nucleotides. Because then that might tell the cell to make a totally different protein because the instructions have changed. So again, found in all cells. So DNA has to be in every single cell. Otherwise, the cell would have no instructions. It wouldn't have anything telling it what proteins to make. Therefore, it would not survive. Here is another picture. So we have chromatin or DNA unwound over here, and it can actually wind up into these beautiful, very organized chromosomes. And your cells do this when the cell is going to actually divide. But most of the time it's found in this kind of unwound spaghetti-like structure because it's actually easier for your cells to access the information then. So it's better most of the time to have it be unwound. And the last of our 4 basic cell structures are free ribosomes. So these are teeny tiny little structures that we're going to find out in the cytoplasm of the cell. So if you can look in this diagram up at the top, it's these little dots. They're kind of light blue. They're hard to see, but they're super, super small. And what they do is they take that information from the DNA. So the DNA is a blueprint. It's useless by itself, right? So if I gave you guys a blueprint to build a house and you don't know how to build a house, that information for you is useless. So DNA is just the instructions. The ribosomes in the cell are actually what take those instructions and create the protein. And in this picture down here, each of these things that look like little beads, those are ribosomes. And they're each making the same protein. And the reason I know that is because they're all kind of stuck together. There's like a little line going between them. That is the message. That's literally the piece that's telling them what protein to make. So all of these little guys are making the same protein, so it just ends up being a little bit more efficient for the cell rather than one ribosome making one copy. If we need lots of copies, then all of these little ribosomes can make it at the same time. And this kind of background right here is just the cytoplasm. Here's a little bacteria cell, a little prokaryote, so you can see there are ribosomes inside of it as well. Not much else though. Right here in the middle, that's all the DNA. So we have DNA and we have ribosomes. That's pretty much it. So we have ribosomes, ribosomes, ribosomes. And our fact is they are made of RNA, which was our second type of nucleic acid. And they're found again in every single cell. So right now, there are 2 questions at the bottom of this chart. I want you to pause the video and take a stab at answering those 2 questions. I just want to see what you guys are going to say. Don't worry too much about it, but give it a shot, and we'll go over it tomorrow. So if you're unsure, we will go over it. I'll make sure that we all understand. But when you're ready, come back here, resume, and we'll move on to the next part of our notes. Okay, so hopefully you took a shot at those 2 questions and you're ready to move on. So as we just saw, and I'll kind of bring it back up, this little prokaryotic cell, or bacteria cell, doesn't have a lot inside of it. So basically it has DNA, it has some cytoplasm, which remember is all this empty, and I'm putting quotes around that empty space, which we know it's not empty, it's that jelly-like substance. And it has these ribosomes, but it really doesn't have anything else going on. And if you look up here at this animal cell, we have a lot more stuff inside of it. So we have to talk about what each of these things do. Why the heck are they in this cell? So all of those structures in that animal cell are called organelles, which means little organ. And they're only found in our eukaryotic cells. So plant cells and animal cells. These are not found in bacteria cells because, again, they're super, super simple. They don't have enough room for all of this stuff. They're a lot smaller than our eukaryotic cells, and they don't need all of this stuff. They kind of live a much more simple life. Here are some pictures of the different organelles we'll find. So this is a mitochondria down here. We have the nucleus, a Golgi apparatus or Golgi body, you might hear it called, a chloroplast, and then we have up here the endoplasmic reticulum. And you guys will learn to recognize all of those things. Every organelle has a specific function necessary to the life of the cell. Just like all of your organs has functions to help keep you alive, and all those functions are different. Each one of these things, this ER is going to do something different than the Golgi, it's going to do something different than the mitochondria, than the chloroplast, but they all work in proximity to each other. They all work very closely together to help keep you alive, and each job is important. No one job is more important than the other. All right, so you don't have to write this down, but we're going to first talk about the organelles of what we call the endomembrane system, which is a system of internal membrane-bound organelles that closely interact. So I want you to try to think of your cells kind of like a factory. They are constantly producing things. So we need a system, kind of like a production line, of how these things get produced from start to finish. And the organelles of the endomembrane system are going to help our cells make and deliver and produce whichever product they need to make. And that might be different depending on the cell that you're talking about. But again, system of internal membranes. Membranes compartmentalize. They divide one area from the other. So really important. The first organelle in the endomembrane system is going to be the nucleus. So in the 2 pictures, it is depicted as purple, and it's generally fairly large. Its job is to separate the DNA from the rest of the cell. So inside of this nucleus, both of these in the plant and animal cell, that's where we're going to find all the DNA. And the DNA is the instructions for doing all of the making of the proteins in the cell. So it's really important that we keep it separate because it's really valuable. If the cell's DNA gets compromised in any way, then that cell can no longer make the correct proteins. So it's important, again, that we keep it separate. It is surrounded by a nuclear envelope, so it goes around the outside. And as you can see in this top picture, you can't really see it in the plant cell, but it has these little pores, these little openings, and that's going to allow material to either enter or exit the nucleus. DNA cannot exit the nucleus. It is far too large and too valuable, but other things do need to be able to, excuse me, need to be able to enter or exit. Okay, you don't have to write down a fact, but it is the command center or the brain of the cell. So again, because it has all the instructions, and it's found in, this is important, both plant and animal cells. The nucleolus, so it's our second box down in our endomembrane system chart. It is this dark spot right inside of each of these nucleus. So that is the nucleolus for the plant and the animal cell. And its job is to synthesize or put together the ribosomes that are then going to function inside of the cell. And if we take a second to remember, ribosomes make something really important. They make proteins. So they make proteins that are working inside of the cell or shipped outwards. Nucleolus, nucleolus. Fact, it's found inside the nucleus, so it's kind of a boring fact, but it's in both eukaryotes, so plant and animal cells. All right, next, let's look at the picture first. So guys, this kind of purple area that looks like it's been cut in half, that imagine is the nucleus. So we're going literally right outside of the nucleus to this pinkish structure right here, this pinkish purple one. This is what we call the rough endoplasmic reticulum, or we can call it the RER for short, rough endoplasmic reticulum. And it has a very specific job. So, do you see these little dots all over it? All those little dots are ribosomes. So we have ribosomes in the cell that are floating out in the cytoplasm, and we have ribosomes that are attached to the ER. And the ones that are attached to the ER specifically make proteins that are going to later be secreted or exit the cell. So this could be like in your pancreas, your pancreas has specific cells called beta cells that make insulin, which is going to help regulate your blood sugar, and that's a protein. So insulin that's going to be secreted from the cell is actually going to be made by the ribosomes that are attached to the rough ER. They're never going to be made by the ribosomes floating out in the cytoplasm. The ribosomes floating in the cytoplasm are specifically for the proteins that are going to stay inside of the cell and work. So any protein made in this rough ER is going to eventually exit the cell. Our fact, it's covered in ribosomes. That's why we call it the rough ER. And it's going to help transport the proteins that it makes to the Golgi apparatus, which we'll talk about in just a second. And the Golgi is going to modify them a little bit. Again, we're going to talk about the Golgi a little bit in like 2 slides. All right, and this is also important. So you could put this under function actually. It's going to help move the molecules that it makes or synthesizes throughout the cell. So it's also kind of a little bit of a transport system inside of the cell. It's like a little highway. So it helps to move the molecules from one part of the cell to the other. And it's going to be found in eukaryotes only. Remember, not in bacteria. They don't have these. And this is actually an electron micrograph. So it's just a fancy picture from a microscope, and it's showing you the rough ER. So guys, these lines are the membranes because that's literally what is building the rough ER. So like kind of folds of membranes, and all these dots hanging onto them are ribosomes. So there's tons, I mean, almost every surface, every space is covered in ribosomes. So it must be really important then for this cell to have these because it's making lots of proteins that are going to be actually secreted from the cell or exit the cell. But then hopefully you notice over here, attached to the rough ER, there was another part that looks a little bit different. So it's just depicted in a lighter color, and it doesn't have the ribosomes covering the outside. So this is the smooth endoplasmic reticulum, or the SER. Its function is going to be that it assembles lipids, so anything lipid-based that the cell needs to make, like steroids, or hormones, or phospholipids, the smooth ER is actually going to be responsible for constructing or assembling those. Another cool thing that it does is that it detoxifies poisons. So anything that you take into your body, like medicine that needs to be broken down, is going to get processed by the smooth ER. So cells in your liver actually have lots of smooth ER because your liver is going to help detoxify your body. Like, that's one of the functions of that organelle inside of you, is that it's going to filter your blood and take those poisons, like medicine, because technically medicine's not supposed to be in your body. It needs to get broken down or metabolized, and it's going to happen in that smooth ER. So the liver cells actually have lots of smooth ER compared to like a muscle cell, which doesn't really need them. Our fact is that it's going to send lipids to the Golgi to be modified and shipped, and it helps move molecules, again, just like its other half, which is the rough ER, so we're helping move molecules. And it's going to be found in eukaryotes. All right, my very favorite organelle, the Golgi apparatus. The Golgi apparatus is located right here. So it's right next to the rough ER and the smooth ER. And it should make sense, guys, because both the rough ER and the smooth ER are going to actually send material to the Golgi, so it should make sense that they're close together. So what does it do? It's the UPS, or the post office of the cell. It's going to process, package, and release proteins and lipids that were synthesized in the ER. And it's going to package these biomolecules, these proteins and lipids, into transport vesicles, which, guys, if we notice, so this is material, say this is a protein that has been made in the rough ER. It's going to move its way through the Golgi, and eventually it's going to kind of be packaged into a little vesicle. So a vesicle is just kind of like a little piece of membrane that's going to surround it and keep that protein safe until it gets where it needs to go. So we don't just kind of release them into the cell, because think of it like a shipping box. We need to have it go to a specific location. So this is going to help ensure that that protein gets to where it needs to go. And it also, the Golgi, another function, it assists in the production of lysosomes. And we're going to talk about lysosomes again in just a second. But it's going to help make lysosomes, which are another organelle for the cell. But again, if you think of it as the UPS of the cell, then you are good. Golgi's right there. It's a flattened stack of membranes. So again, all these things are covered in membranes. That's the point. But this one's kind of easy to recognize because it has this kind of very distinct flattened look to it. And it's going to be found in plant cells and animal cells, as has all of the parts of the endomembrane system that we've talked about. Here is another kind of electron micrograph. So again, fancy microscope picture. All of these lines right here, those are part of the Golgi. And these guys right here are little vesicles that have been produced from the Golgi that are going to go and do their jobs. They're going to send their protein out of the cell to where it needs to be. Okay, so vesicles, we just talked about them with the Golgi. They're going to help transport those cellular products to the final destination, which most likely, guys, is going to be outside of the cell, because that's what these products are generally destined for, especially if it's a protein and it's being made in the ER, and it's going to be sent to the Golgi, and the Golgi is going to eventually ship it out of the cell. And these guys also help move materials between the ER and the Golgi. So if we look here, they're not actually connected. The Golgi and the ER aren't connected. So when a protein moves from the rough ER and it needs to go to the Golgi, it's actually going to be packaged into a vesicle to be shipped to the Golgi. The Golgi is going to grab it, modify it, make all the changes that it needs to make, and package it into a new vesicle to be shipped out for its final destination. okay? And it's, again, found in eukaryotes. So if you look, this is just kind of a beautiful picture of all that's happening here. So here is the nucleus. Here is the ER right around here. There's a bunch of vesicles. They go to the Golgi. The Golgi then ships out the final products in vesicles, very similar to these vesicles. And these guys, these proteins, probably getting secreted or pushed out of the cell because they have a function that's going to need to be bigger than just staying inside of the cell itself, kind of like that insulin. So insulin eventually, once the pancreatic cells make it, gets released into the blood. Okay, so how do these organelles work together? You should have on your chart an organelle interaction column. We'll go over that. I'll make sure that everybody is good with that, but let's just kind of go over this. So you don't need to fill out that section of the chart just yet. Proteins are made by the rough ER. So if we have a protein getting made by the rough ER, it's going to get sent to the Golgi using vesicles, bubbles of phospholipids, again, just bubbles of membranes. The Golgi then modifies the proteins and sends the updated proteins to the cell membrane by using vesicles, again, right here. So we have rough ER stuff packaged into vesicles, Golgi, and then out. The vesicles then are going to merge with the plasma membrane and release the proteins outside of the cell. So again, this cell is not making those proteins, those particular proteins for itself. They have some other job to do outside of the cell. Okay, so lysosomes. Lysosomes are made by the Golgi, and they're really important. I want you, when you think of the word lysosome, to think Lysol. So Lysol is a cleaner. The lysosome is also a cleaner. The function of the lysosome is to use digestive enzymes, remember, they are proteins, to break down old organelles, bacteria or other foreign material, or sometimes food. So the lysosome is just breaking things down. And they're kind of hard to tell what they look like. They're just small and circular. They're going to look very similar to a vesicle. So if you ever are in doubt of what something is pointing to, just ask me because, again, lysosomes and vesicles look very, very much the same. Fun fact, they are called suicide sacs because when a cell needs to, kind of live out the end of its life, because cells do this, we recycle old cells all the time, the lysosomes in the cell will actually open and they will release their digestive enzymes into the cell, and the cell will be digested from the inside out. It's called apoptosis, or programmed cell death. And guys, be careful, these are found in animal cells only. Plant cells do not have lysosomes. So this is our first organelle of the endomembrane system that is not found in both plant and animal cells. So remember, only animal cells. So if lysosomes don't function correctly, you wouldn't think that their job is very important, but it actually can lead to some really horrific things. So in Tay-Sachs disease, which this little boy in the picture has, the lysosomes, especially in nerve cells in your brain, don't function correctly. So waste actually builds up inside of those cells, and the babies don't actually live past the age of 5 because it's a degenerative condition. As the waste builds up, it gets harder and harder for their brains to function. Those cells become toxic. They don't work. They die, and then the child will die. So it's actually really sad. So it's a disease, a genetic disease, where these lysosomes, again, don't function correctly. So this is just to hit home that every single organelle inside of these cells is important and does a really, really important job. And no organelle is more important than the other, because when one stops working, it can lead to an effect much larger than you would think. All right, so the vacuole is, this should say endomembrane, so it still should be part of the endomembrane. The vacuole, if you look in this cell, it kind of looks like a pool. So it's very, very large in our little plant cell here, and it's for storage. It's going to store our food. It can store water, it can store waste, it can store proteins, it can store carbs, it can store pretty much anything. And it's going to help maintain homeostasis. So in our plant cell, water balance is going to be really important. So when a plant has access to a lot of water, it's going to take it in and it's going to store it in this vacuole. So that way, when there is a time when the plant doesn't have access to fresh water, it has this extra water stored up. So that's, again, super important. It's going to be found in both plants and animal cells, but you do want to write down that plants have a single large vacuole, which is like this little swimming pool right here. Animal cells are going to have several small vacuoles. We don't have that giant vacuole right in the center. So guys, actually that's where we are going to end for today because I don't want to do too much for you guys doing this at home. I really appreciate you taking the time to do it at home. We'll finish up because there shouldn't be too much left. So we'll finish up the rest tomorrow in class. And again, thank you so much. I will see you all tomorrow. Have a good rest of your night.